Okay, so as announced already, I would like to give you our ideas about automation of immunofluorescence. We talked a lot about ANA today, so which could be found in many diseases. What maybe is a little bit interesting also is to have a look at the histology history. So ANA was described first in 1948 in SLE patients, and the indirect immunofluorescence came up in 1957 mainly on red liver tissue. So, and as you know, red liver tissue maybe is not that sensitive than Hep cells. And this also maybe explain a little bit why we still some use 1 in 40 as a screening assay, because if you use tissue, maybe 1 in 40 is the correct cutoff value. So also the criteria was introduced at that time. Then these double immune diffusion assays came up for monospecific confirmation of ANA patterns in 1966, and there was a huge step in 1975 when ANA flipped from the tissue to the Hep cells. And the Hep cells are much more sensitive than the tissue, and you could recognize much more pattern in a much better way. So, and this is really a milestone in the history of ANA diagnostic. Then many confirmation tests came up, like the Western blot in the 80s. In the 90s, screen ELISAs came up. 98, a lot of line blots came into the market. And in 2005, many multiplex assays start for ANA diagnostics. With all the drawbacks multiplex assays have, you may are aware of the meeting in the United States from the ACR, which later on recommended to use the Hep cells for ANA screening and not a multiplex assay. But of course, one drawback of immunofluorescence is that it's time-consuming, it's not standardized, the reading is quite difficult, you need a lot of experience. But then in 2010, automated systems came up. So, and these automated systems are now able to take automatically pictures of the Hep cells and evaluate the images and give a pattern. But beside the automated systems, there are many more advantages you can use. One, of course, is the automated incubation, because many IFA workplaces are still running manually, and this, of course, also leads to a less reproducible result compared to automated systems. We have a software which we call Eurolab Office, and Eurolab Office link the incubation and the evaluation scheme. So everything is modular, so you don't have to take it all. If you just want to start with a microscope or just with the automated incubation, that's pretty fine. And Eurolab Office links to your list system. So we import all the patient data and the requests to Eurolab Office, export or create work lists for the incubation with the dilutions, the different conjugates, export that to the Sprinter system and or to the automated incubation system. And this is exported to LIS and given to the microscope then, so that the microscope exactly know which patient is in which dilution with which conjugate on which slide and well. So, and this enables us to have a complete paperless workflow. So you don't have to fill in any paper anymore. You can do everything automated. If you need paper, we can print paper. That's no problem, but you don't have to, because also lot numbers, expiry dates, everything is tracked within the system. So one example for incubation is what we call Sprinter XL. Sprinter XL is able to run 240 samples, 30 slides. It's a 2-arm device with 4 needles, so also the time management is pretty fast. It handles barcodes, so our slides are barcoded, so we have full traceability here. The washing is done by flooding, so it's not really putting a droplet, aspirated droplet, but it flushes the complete slide, so the washing is very efficient and also not that time consuming. And it could handle immunofluorescence and ELISA. This is for smaller labs. This one is for bigger labs, what we call Eurolab Workstation. It's still under development. It's a completely flexible system where you have a liquid handling device and you have your slides on a rack, and this rack then is taking over to the washing unit and later on also to the microscope. For the washing, we developed a completely new system which we call Megite. So here you have your slides on a tray upside down. So this is a slide, or illustrate the slides. Here we have the substrates, for example, the Hep cells. And this washing unit then flushes the biochips from downside. And it flushes 50 wells in one step. So and the step roughly takes 20 to 30 seconds. So it's very fast and efficient washing. And you don't have any cross-contamination between the different fields, which you from time to time have. If you have a strong positive and you pour your wash buffer about the other well, you have a weak centromere on well 2, but in reality it's well 1. So this couldn't happen here. I mentioned already the automated system. The system automatically focus and image the Hep cells. All the data management is done by Eurolab Office, also the connection to your list system, so reports later on could just be exported to the list system. And we have a slide identification by matrix code, which enables us to fully trace the patient. We have a very rapid autofocus, which we do in transmission light. That means at that point of time, we have no bleaching. So autofocusing is a pretty long process, takes you roughly 15 seconds. And if you do that already with blue light, you have bleaching effects. And when you take your picture, your image is already dark, or at least 2 titers below. That could happen. That couldn't happen with our system because we use transmission light. And we just switch to the blue light when we take the picture for 0.2 seconds. So the throughput is roughly 80 seconds per well. And we could handle different objectives. So for Hep cells, we do the 20-fold magnification. For tissue, we mainly use the 10-fold. For Crithidia, for example, we use the 40-fold. And you can take this microscope also completely as a manual microscope, means you can also visually read without using the software. We have a large capacity. You can load your slides. So we have 10 well slides and 50 well slides, which you can load into this kind of magazine. And this magazine you attach to the microscope. And then you run, just press the start button, and 500 wells will be automatically imaged and read. So as mentioned, the system is able to run ANA, but also other systems. I would like to start with ANA. We already had a look at that we have the 7 most common ANA patterns which we report, and these are the competent level patterns of ICAP. So you may remember from my first talk that there were some summarized in one pattern, but this is what also ICAP is doing on their webpage. So combination of patterns could be found here. We have a titer estimation. We use counterstain, which is pretty important for negative cases, because otherwise you don't know, did you do a picture in the wrong level, because then it's also black, or it's just a negative sample. So that's why counterstaining is very important to verify the autofocus process. We also have what we call batch-wise verification of all negative samples. That means we have a drop or scroll down menu where all the negative samples are listed by brightness. So the brightest negative is on top, and then you just scroll down and say, okay, I agree, or if you disagree, you just activate this sample, and then this sample went into the positive list. And what we're also doing is we give a result per patient. That means we don't give a result for well 1, 2, and 3. If you have different dilution on different wells, we combine these information in one final result. This is also different to the competition, which gives you a result per well, and then you manually have to merge these results. This is done automated here by the software. So we looked already into that. And this is what I meant when I said we have the 7 main patterns. So you see here homogeneous, and also the speckled group is one pattern we report, the centromere, the nuclear dots is one group we report, and we also give one result for nucleolar. So we don't distinguish homogeneous, clumpy, and punctuate here. So we had this slide already. So further key facts or advantages: you need no darkroom anymore, so you can work from different workspaces. So everything which is connected to the server, each computer which is connected to the server could be used to validate the results later on. We have a feedback function, so if you have any doubts, you just click a button, and then the image, without any patient information, just the image is forwarded to our specialists in Germany, and they will feedback within 24 hours what is their opinion about this image or this pattern. We have 3D control for live microscopy, so if you want to take additional pictures or have a look at slightly different levels, you can use a manual control, and you can also use the oculars. Furthermore, we have a CLED, means controlled LED, and not an HBO bulb anymore. These CLED give constant light intensity for at least 50,000 hours. Means you don't need to replace the bulb anymore, no beam adjustments anymore, so no maintenance anymore at all. So it works for 50,000 hours without any maintenance. We identify the mitotic cells and could highlight that. I mentioned already the pictorial atlas we have, and you can add your own patterns to a list of the automated patterns. So here these are the patterns we report, but we also can add some manual, and this is customizable. So if you want to have additional patterns, we just can define that, and you directly can activate them if you think it's positive. So I mentioned the counterstaining, which we are using. By the counterstain, we know where the cell is located, and we do a cell template, and in these white areas, we look for the specific green fluorescence. Means also, if we have unspecific fluorescence in the black areas, it doesn't affect our evaluation, because we even don't look there. So we just look in the cell template areas. These are the images or the patterns we report. And this is based on more than 100 different feature values. So it's a very complicated algorithm, and 150,000 cell pictures from routine samples are in the background as a training database for this software. I mentioned already the pictorial atlas, which you can use to train your own people and also to have a standardized cutoff value. And how does the software now evaluate if this is a homogeneous or a speckled pattern or whatever? So what we are doing is we have, in this case, as an example, 50 reference samples for centromere, and these 50 reference samples form out a kind of cloud in a 3-dimensional room, or because we look at 170 feature values, it's a 170-dimensional room, but nobody can imagine that, so that's why I put it just in 2D. So we have a cloud for centromere, and from our speckled reference images, we have a cloud for speckled, and then we just put your sample in these clouds and see what are the 9 nearest neighbors. And in this case, you see your sample have 7 green, so means speckled points here, and 2 blue, which are from the centromere cloud. So your sample seems to be more speckled than centromere. And the software also reports a confidence value, means the more that your sample goes in this direction, the more sure the software is that this is a speckled pattern, and the other way around in this direction would be more sure that it's a centromere pattern. And this is what we're doing for each single pattern. So we look for how many percentage. This is a homogeneous pattern. This is a nuclear pattern. This is a speckled pattern. And for this reason, we also could report different patterns in parallel. So if it's above a certain cutoff, we report it positive. And that's why we also are able to handle mixed patterns. So we don't have predefined mixed patterns. We are really looking for the patterns. So this is a negative list I mentioned. So here you see all the negatives. We have the red image here aside the green image, because then you can clearly identify, okay, the autofocus level was correct. And if you disagree, you just deactivate the negative checkbox and it automatically will go into the positive list. This is a positive list. So you see you have your different dilutions here of this sample, so merged to one final result. So here are the different dilutions. We also could add, for example, a liver image. If you have a Hep-liver combination, we could also image your tissue beside the Hep cells. You can magnify all dilutions. You can scroll within the image. And this is the report box. So the computer result is in red. So this is what the computers say. Okay, it's centromere, titer is 3,200. You can put your visual result in addition, and this is the final result, which is later on transferred to the list system as a result. You have also here additional results from the ELISA or blood, so that you directly have all information of this patient. And what I already mentioned, so these are the patterns we report automatically, and these are the customized patterns which we could add if you want. So we also have that for Anchor. For Anchor, we have different products. So we have ethanol and formalin-fixed granulocytes. We have Hep cells with granulocytes in addition to exclude ANA reactivity, because ANA could mimicry a P-anchor pattern. And we have monospecific dots, which purified antigen to confirm the result of the granulocytes. And this is how it looks like for the Anchor classifier. So here you again have your different dilutions, and then you have the different biochips, which is combined in one well. And on our slides, and if you just have ethanol, just one image will appear here. If you have a 6 biochip combination, all pictures will be displayed here. And then it's the same, so you have your computer result, and you can have your official result in the end. You also could put unofficial remarks, so if you want to give a note to your technician, so please re-incubate tomorrow, or do a different dilution, or look at the ELISA, you just can put it here, so, and then it's just visible for your technician, but not visible in the report. Later on, you export to the list system. Also here we are working with counterstain, so to exclude the eosinophils, so you see that they are stained differently, and also here we use a cell template, and we just use the granulocytes which are single and not coupled, or the eosinophils we even don't take into account. And then we report C-anchor, P-anchor, a group which we call non-anchor associated anchor, or AAV anchor, which means it's an atypical anchor or P-anchor, which could mimicry an ANA. Then we report the dots, positive or negative, and of course also here we have combinations of patterns. Criteria is pretty much the same. So also here, if you do different dilutions, they will be displayed here. You can magnify one image, and then you just have a positive, negative result for your criteria. If you disagree, you just deactivate the box and activate the negative box, and then it's negative. I think in this case, nobody will do that. If you disagree with the titer, you click on the titer and could choose one of your titers which you think, or which you estimate from that. So also here we use counterstain, and the classifier detect the kinetoclast beside the nucleus and the basal body. We could image tissue, so this is just one example. Of course it's negative, so it's maybe not the best example, but also here you can have liver, kidney, stomach, in addition Hep cells on one biochip, and it could be imaged automatically, but then you manually have to evaluate the images. So this is not automated by the computer so far. We also have a lot of transfected cells. I don't want to go into detail here, but we have a lot of transfected cells in our portfolio. For example, we've heard today about NMDA receptor already. Zika is also one, which we have in our mosaic. And these could also be automatically evaluated meanwhile. That means they are imaged automatically, and then the software tells you it's positive or negative for all these transfected cells. So just as a conclusion, so we have some which are completely automated, or where the evaluation is completely automated, and some are CE, or most are CE approved, some are even FDA approved. Anchor and double-strand DNA is just under approval for the FDA, and we have some where we can do the imaging and the achieving, like anomasium, neuronals, liver, kidney, stomach, and further on. I think because my time is pretty much over, and Jan has some results as well, I don't want to bother you with that. Maybe just some key points. As mentioned, 50 barcoded slides could be put in, and you have a fully automated workflow without any paper anymore. We have installed over 200 systems in 27 different countries. So biggest customers are Brazil. So as mentioned, they run 2,000 ANAs a day. Japan also run roughly about that. I think they have 4,000 ANAs a day. And also in the USA, it's quite interesting because there the labs are not centralized, so they are spread all over the countries. So big companies like Quest or AAUP are interested that the result in New York is the same than the result in LA, and this is what they like to ensure with our system. Okay, so with these automated systems, we really eliminate the IFA drawbacks, which is time-consuming incubation, time-consuming evaluation, and subjective evaluation. So paperless workflow I already mentioned, and this was just a short introduction into the possibilities you have for automated IFA. Thank you very much.